The overall goal of these procedures is to objectively quantify honeybee colony status by using adult bee masses and brood and food resource areas to thus measure colony health in response to agrochemical exposure.
This method can help answer key questions in bee science, such as colony-level effects of exposure of sublethal doses of agrochemicals to honeybees. The main advantage of this technique is that adult bee masses, brood areas, and food resources are rigorously measured rather than visually estimated, and that a record is kept to double-check observations.
We have developed organizational systems to minimize disturbance of the colony. There is a learning curve to the process that is important to work quickly and efficiently.
It is helpful to have this method demonstrated visually due to the complexity of the procedure. Weighing the components of the hive and photographing the frames are done systematically in order to be less intrusive in the colony.
Always conduct hive evaluations when ambient conditions are suitable for working with bees. To begin, place a temporary empty hive box on a flat surface next to the hive, such as on an unused hive cover, so that the queen can be collected if she falls out. Then, weigh the hive lid, and if present, the inner cover. Next, use a spatula to remove and weigh any protein supplements or pest treatments that are resting on the frames. Keep those materials and replace them on top of the frames at the end of the evaluation. Now, note the order and orientation of all the frames by marking them with indelible ink. Then, into a temporary